Essentials in molecular biology. Learning objectives. 1. Connect gene structure to protein production via cell central dogma. 2. Understand the structure of DNA molecules and the organization of genes in a DNA molecule. 3. Comprehend gene expression and its regulation. Based on the most recent estimates, there are around 30 trillion cells in the human body, which all work in harmony to carry out all the basic functions related to survival. They all are categorized into 200 different types. Despite their structure, size, shape, and function differences, cells all work together to keep the body running efficiently. They also can contain different organelles, however, there is some similarity between them. One of the most important similarities is related to the flow of information inside the cell. Like in other cells, in human cells, the information stored in the DNA structure and can pass to major functional units, proteins, by adapter molecules called RNA. Proteins have the same information as DNA but in another language. This flow of information from the DNA structure to protein structures is a fundamental concept known as the central dogma of molecular biology. Thus, the information should transcribe to RNA structure and then be translated into another language with a different alphabet. Let's have a closer look at the inside of the cell. In eukaryotic cells, the main part of the DNA is stored inside the nucleus as some separated molecules. In the human somatic cell's nucleus, information is stored in the structure of 46 molecules of DNA, each of which composes a chromosome structure, with a defined length, shape, and piece of information. Each chromosome is composed of long DNA molecules that are folded around specific proteins, forming a compact structure. Just by removing the proteins that are involved in chromosome structure, the intact DNA molecule becomes accessible. Let's have a closer look at the DNA molecule. The two strands of DNA bond together in an antiparallel format, with each strand extending from the 5' end to the 3' end. In a DNA molecule, there is a sequence of various genes, each containing unique information required for producing specific functional products. Intergenic sequences are regions between genes, which don't code specific products. Traditionally, these sequences were thought to lack specific information. However, in recent years, it has become evident that they play a significant role in storing valuable information. In a closer look, each gene on a DNA molecule has a specific sequence of nucleotides, as well as distinct starting and ending points. The start point of a gene indicates the direction of RNA polymerase activity, determining the orientation of transcription. It also helps identify which strand of the DNA molecule serves as the template for RNA production. The promoter sequence provides important information to the RNA polymerase, indicating the specific location where transcription should begin on the gene. It serves as a signal for the initiation of gene expression. The RNA polymerase binds to the DNA by recognizing the promoter sequence. Once bound, the RNA polymerase proceeds to produce a complementary RNA transcript using the template strand of the gene. This process allows for the synthesis of an RNA molecule that carries the genetic information encoded in the gene. Finally, when the RNA polymerase reaches the end of the gene, RNA molecules that carry the same information as the gene are produced. These RNA molecules have a specific 5' to 3' orientation, mirroring the template strand of the gene. The resulting RNA for protein-coding genes contains a start codon, AUG, and one of the stop codons, UAA, UAG, UGA. The start codon serves as a signal for the ribosomes to initiate protein synthesis, while the stop codon signals the ribosomes to terminate the translation process and release the synthesized protein. These codons play a crucial role in determining the proper reading frame and ensuring the accurate production of proteins from the RNA molecules. Based on the location of these codons, the RNA sequence can be divided into 3 parts. The 5' untranslated region, 5' UTR, is located before the start codon, which does not encode any part of the protein. Open reading frame, ORF, encompasses the region between the start codon and the stop codon. It contains the nucleotide sequence that encodes the amino acid sequence of the protein. The 3' untranslated region, 3' UTR, is located after the stop codon. In eukaryotic cells, genes can have an extra feature. Let's have a closer look at a typical eukaryotic protein-coding gene. In the gene sequence, there are regions of DNA that directly contribute to the production of proteins. These regions are called exons. Exons are interspersed between the non-coding regions of the gene known as introns. The presence of introns in the gene sequence allows for additional complexity and regulation in gene expression. Introns may also contain regulatory elements that influence gene expression and other important cellular processes. During gene expression, the entire DNA sequence of a gene is transcribed into a precursor messenger RNA, pre-mRNA, molecule. This pre-mRNA molecule contains both introns and exons. During the process of RNA processing, known as splicing, the introns are removed from the pre-mRNA molecule, and only the exons are retained. Joining exons together, a mature mRNA molecule forms that contain only the protein coding information. On mature mRNA, there is a start codon and a stop codon. The sequence between these two specific codons is translated by the ribosome, and both UTRs in both ends stay untranslated. During this session, we discussed the flow of information inside the cells, which is from DNA to RNA and then to protein. We highlighted the structure of DNA molecules, gene structure, expression, and regulation.